Patricia King is a graduate of Wheaton College and received her JD from Harvard University. She's the Carmack Waterhouse Professor of Law, Medicine, Ethics, and Public Policy at Georgetown Law School. She's also an adjunct professor in the Department of Health Policy and Management at Johns Hopkins School of Hygiene and Public Health. After graduating from law school, she worked as a lawyer in the federal government, primarily in civil rights. She was Deputy Director of the Office of Civil Rights and Special Assistant to the Chairman of the EEOC. Bet you don't know what that is. Equal Employment Opportunity Commission. She also served as a Deputy Assistant Attorney General in the Civil Division of the Department of Justice. Professor King's academic research has focused on the intersection of law, medicine, ethics, and public policy, with an emphasis on biomedical science. At Georgetown, she teaches courses in family law and bioethics in the law. She's a member of the American Law Institute, the Academy of Medicine, and a fellow of the Hastings Center. She's served on several national and government advisory groups studying biomedical issues ranging from genetic research to experimentation on human subjects. Her work in the field of bioethics has included service on the HEW Recombinant DNA Advisory Committee and the President's Commission for the Study of Ethical Problems in Medicine and the National Commission for the Protection of Human Subjects of Biomedical Research, and the Ethics, Legal, and Social Issues Working Group of the Human Genome Project. She has been a fellow of the Harvard Corporation and chair of the Board of Trustees of her undergraduate alma mater, Wheaton College. Today she will speak on gene editing, the ethical dilemma.
Thank you very much for that wonderful introduction. I feel like a fish out of water, having listened to all of the speakers before me, but I'll do my best. I start with one caveat. I am not a scientist, but you cannot do what I do without spending a lot of time around scientists. So bear with me. And the science in this area is hard, and I hope I have gotten what I need down pat, but we will see. So my career parallels roughly what I'm going to talk about today. Since the beginning of molecular genetics, scientists and people like myself have been concerned about the safety and ethical implications of human genetic engineering. The term itself was defined in 1965 to include a wide range of techniques by which scientists can add genetically determined characteristics to cells that would not have otherwise possessed them. In the 1970s, scientists figured out how to isolate DNA sequences from one species and attach it to another. Thank you. The recombinant DNA controversy that followed led to a moratorium and a great deal of controversy, and the creation of the Recombinant DNA Advisory Committee that created guidelines for the conduct of this research. And those guidelines were applied to all government-funded research, and in the private sector, companies voluntarily agreed to abide by them. In 1982, the President's Commission for the study of ethical problems in medicine and biomedical and behavioral research, of which I was just briefly a member, was established by Congress and issued a report called Splicing Life. This report was in response to a letter received by President Carter in 1980 from the General Secretaries of the National Council of Churches, the Synagogue Council of America, and the United States Catholic Conference. In the wake of a Supreme Court decision that allowed the patenting of new life forms, these organizations were calling on President Carter to remedy the lack of adequate oversight or control among governmental bodies by providing a way for representation of a broad spectrum of our society to consider these matters and advise the government on its necessary role. At that time, and this was the 80s, the President's Commission understood its role as stimulating thoughtful conversation and a long-term discussion. In its report, it raised what some have called, and they call, the Frankenstein factor, and they did so deliberately in order to convey the level of public uneasiness about how gene splicing might change the nature of human beings. That report also raised a number of ethical and social concerns and called for continued research with respect to human genetic engineering to improve human health with great regulatory oversight. So today, some 30+ years later, society finds itself in a position where it urgently needs to revisit the scientific, ethical, and social implications of human genetic engineering. With dramatic breakthroughs in this field, we must educate the public about these developments, clarify and resolve issues raised by complex technologies that facilitate gene modification, and identify the pertinent social and ethical issues and determine what, if any, regulatory oversight is needed. So what has happened? Well, in April 2015, in an international first, a group of Chinese researchers led by, I don't know how to pronounce his name, Junjiyu, I believe, I hope that's correct, Huang, reported an experiment that utilized a gene editing technology called CRISPR-Cas9 to modify genes of a human embryo. The researchers had deliberately chosen to only use embryos that were not viable, so thus they would not be implanted. They were attempting to eradicate a genetic disease, human beta globin, and they were trying to see if they could modify in the embryo's germline. If it was possible or became possible to modify a human germ cell, an embryo germ cell, then it might be possible to disrupt transmission of the disease to the next generation. The scientists' work was astounding, significant, because in principle, it made us all aware that we were close to the point where we had to think about trying to disrupt genetic diseases by changing the germ cells in the embryo. This experiment wasn't completely successful, but it also represents a major crossing of the boundaries in human genomic engineering, and thus posing many challenges for science and society. I should point out that since the first report, there's been a second report of another Chinese research team doing similar experiments, but trying to prevent transmission of a different disease. Now, assuming that the safety and efficacy concerns connected with gene editing technologies with embryos are met, we've got a critical issue in front of us, and that is whether efforts to treat or prevent severe genetic diseases with appropriate guidelines is a responsible use of human germline engineering. While this work offers the prospect, obviously, of eliminating genetic disorders in humans, it also raises numerous questions. Foremost, will we change the nature of human beings? I seem to hear a theme running to some of the presentations today about the nature of human beings. But if there are what if there are adverse consequences for future generations? Fixing genetic variation that causes a rare disease is probably a laudable goal, but is there a consensus that distinguishes normal variation and disability? How should we think about genetic defects like deafness and dwarfism? Do we put them in the category of disability, something to be treated? Or do these two diseases, for example, just illustrate the range and variability of human diversity? And we should ask, I think, clearly whether our therapeutic goals, which are important, could be achieved in some other way, some way other than trying to avoid transmission to the next generation. And some would argue that we could modify DNA sequences in pluripotent cells derived from somatic cells, where there's currently broad agreement that gene editing is appropriate. But the research in 2015 not only was a step across the line that dealt with possible therapeutic interventions, it also opened up the prospect of something else, something that my students are absolutely enchanted by, and that is the prospect that we will use genome editing techniques and go beyond therapeutic modifications and attempt to enhance human beings. And the question would then be, should we use these techniques for that purpose? Would it be wise to do so? And if we were to try, then it would raise very serious questions about the child that might be produced, what human traits should be selected as being preferable, and is the distinction between genetic therapy and genetic enhancement all that straightforward anyway? Is editing out heritable traits from the human population really eugenics in another guise? So we have discussed these issues for many years, as I've indicated. The change is that the new editing technique, CRISPR-Cas9, promises to accelerate some of these possibilities, and thus there's a need to start thinking again about what are the consequences and how far do we go forward. Now, the researchers in this Chinese experiment used CRISPR-Cas9 which is shorthand for clustered, regularly interspaced, short palindromic repeats CRISPR-associated system, known as CRISPR-Cas9, that allows researchers to introduce or correct genetic mutations in an organism's specified segments of DNA. CRISPR-Cas9 is a technique that employs a part of an organism's bacterial adaptive immune system that recognizes and removes foreign DNA from the bacterial genome. The work of people like Jennifer Doudna and others in the last several years has resulted in the creation of CRISPR-Cas9 and an explosion in the different arenas that it can be used. It's not the only gene editing technique. There are others. But as of now, CRISPR-Cas9 is considered the best and the most versatile. It's simple. It cuts and deletes precisely. It's easy to use. It's inexpensive. It works in all living organisms, from plants to mosquitoes and humans. Indeed, the attributes of CRISPR-Cas9 are so astonishing that they make the thought of trying to regulate use of this procedure difficult, because a person doesn't need expensive equipment or years of training, as demonstrated by one of the reporters in Science magazine, to figure out quickly how to do this. It's being used, for example, something that I care about, with sickle cell disease, in an attempt to still try to find some way of ameliorating that disease. It is also being used in the broad arena that we would call personalized medicine. Now, the Chinese experiment used embryos, obviously, but any kind of research just on the genes or on the germ part, the germ gene cells of embryos, is controversial. It has been so in the United States since 1992. I know because I chaired the NIH committee that was to establish the U.S. policy on embryo research, and it failed because it stirred quite a political storm. Congress had in '93, finally, after 20 years, explicitly permitted NIH to do research on the human embryo. And the report of the committee that resulted, obviously, was only adopted for a while. In 2000, Congress reversed itself and passed the so-called Dickey-Wicker Amendment that prohibits federal funds for creation of human embryos and research on embryos in which a human embryo is destroyed or subject to risk of injury or death. The obvious problem with, and the controversy around doing research on the human embryo, is because it is a human embryo, and the issue is, what is its moral status? Is it a person? Is it just like all other human tissue? Or does the human embryo fall in some mid-level category where it is greater, deserves greater respect than mere tissue, but less than that afforded human persons? The international community is equally divided about research on human embryos, so this is not going to be a national issue by any means. Some countries prohibit the research, Other countries like Austria and Italy permit research not on embryos, but on cell lines that have been generated in other countries. Still other countries explicitly endorse and fund human embryonic stem cell research. The UK is one such country and probably has the most comprehensive regulatory approach of any of the countries that are currently involved in research on embryos and other forms of research. This past February, HICFA, the British regulatory system, approved an application by a developmental biologist to use CRISPR-Cas9 in healthy surplus embryos. The researcher is interested in early human development and intends to only alter genes that are active in the first few days of life after fertilization, and then the embryos will be destroyed. The science community, I have to say, has been splendid in its responses to these developments. There was a group of scientists that met in Napa in 2015 prior to the publication of the Chinese paper. They drafted a statement, which was subsequently published online, that the potential safety and efficacy issues arising from the use of the technology CRISPR-Cas9 must be thoroughly investigated and understood before any attempts at human engineering are sanctioned, if ever, for clinical testing. The scientists stated that steps should be taken to strongly discourage any attempts at germline genome modification for clinical application in humans until there was an adequate international discussion of human genetic engineering in which genetic modifications would be heritable. This past December in Washington, scientists, ethicists, legal experts from 20 countries around the world met in an international summit in Washington, and they too issued a position statement, or one was issued by the organizing committee, which didn't actually condemn modifying a gene in embryos, but suggested that many ethical and safety issues had to be resolved first. So where we are is that the science community has responded admirably, in my view, and they're trying to grapple with these issues. And the reason I went through some of those events is to make it clear that I'm not anti-science, and I respect scientists. But there is an important question that remains, and the question is whether scientific self-regulation alone is adequate to grapple with the complex questions raised by modifying the human gene line. I believe that the ethical and social implications of scientific knowledge go beyond what scientists can competently treat within their areas of expertise. Moreover, since the 1980s, science has changed. It's more insular. It's more driven by profits. There's even a patent dispute about who should get the patent to CRISPR-Cas9. So my point here is that we need to make a transition from a predominantly scientific exploration of these issues to a broader social one. And my starting framework for that transition would be agreement on a framework of ethical principles, much like that used by the National Commission in Human Subjects Research many years ago, because a framework of principles to guide these discussions needs to be, in my view, quite broad. And I think the work has been done in terms of the framework. A Presidential Commission for the Study of Bioethical Issues, in a report on the ethics of synthetic biology and emerging technologies, which issued a report under the auspices of the president, actually, of the University of Pennsylvania, attempted, with respect to emerging technologies, to do exactly this. So briefly, because it has to be brief, they came up with 5 guidelines. 1, public beneficence, or that we would all have to act to maximize public benefits and minimize public harms. 2, responsible stewardship, or the obligation to proceed prudently for the betterment of all, especially those who cannot represent themselves. And 3, intellectual freedom and responsibility, or the creative spirit of scientists, must be promoted and protected, but at the same time, the responsible science should reject the technological imperative that the mere fact that something can be done means that it should be done. 4, democratic deliberation, or an approach to collaborative decision-making that embraces respectful debate. I'm beginning to wonder if this can happen in the next 4 years, of opposing views in order to strive for mutually acceptable reasons to justify policies to be adopted. And 5, justice and fairness, or reasonable efforts on the part of both individuals and institutions to avoid unjust distribution of benefits, burdens, and risks that technologies bring. This 5th point is actually a point that is often dismissed or lightly treated with respect to emerging technologies, but in the world that we are coming to know better, it may turn out to be one of the most important. So the next step, in my view, would be to establish a neutral national forum. I would have said a bioethics commission, but they're established by presidents. So it's got to be a neutral national forum or an appropriate international body that includes scientists and science organizations with the charge to conduct a broader and more diverse discussion of genome editing. And for me, that would constitute a prudent step forward. And I think I should close here by saying that in England, where they've just approved mitochondrial donation therapy, which is not in the same category that I have been discussing, you probably know that advance by the term 3-parent babies, because it involves the contributions from 3 adults to prevent the spread of mitochondrial disease, that their oversight system required 4 years, and this is a relatively small area, 4 years to review safety, efficacy, ethical issues, engaging in public consultation, reviewing draft regulations and recommendations for future research. I think thinking about crossing boundaries in human genetic engineering deserves no less. It's probably not an appropriate process for many issues, but I think it is appropriate for this one. Thank you.
Extremely important issues. Herb?
So, Steve Lippard from Cambridge. That was really interesting. It's a shame that Jennifer didn't actually show up to give the talk.
She was going to give the talk, and then I was going to get to say what I really wanted to say, but it didn't work out.
Not that we didn't enjoy Mike. We all enjoyed Mike. So I'm aware of some work where people are trying to to find a vaccine to prevent cancer. And it's a vaccine that would be given to pregnant women, so it would be done in the developing fetus. How do you feel about that? Do you think that is been with the same sort of genetic modification that you've been discussing, or do you think that would be in a separate category?
Well, it raises an entirely different area of consideration. As you probably know, under our existing guidelines in the United States, I don't know the rest of the world about this question, we don't do much research on pregnant women. And one of the reasons we don't do much research on pregnant women is because of our concern about the fetus. And even though there are women's organizations that are trying to change this, they have not been successful so far. So we have an additional obstacle, even though it may be for a very good purpose in this case, that we're going to have to consider and somehow convince the people to change the regulations to allow that in this country. In other countries, I think they face the same kinds of dilemmas that I tried to raise here. It's not a question that doing research that would lead to a therapeutic goal is a good thing. The question is, what other considerations should you also consider at the same time? Okay, thank you. The goal is to go to humans, but in this country, because this is an issue near to my heart, in this country you don't go to humans with government-funded research. You might be able to do it with privately funded research.
Erwin Shapiro, Cambridge, Mass. I was interested in the fact that for these ethical controls of, say, CRISPR-Cas9 experiments and things like that, the results are clearly could have international effects.
Sure.
Why is there any effort to organize, say, under the United Nations or something like that, guidelines that will be adhered to internationally?
Well,
I realize that might be very hard to accomplish, but it's no less important, perhaps.
Well, the meeting in December, as I mentioned, was one effort to do that, and the meeting was between the Americans, the British, and the Chinese. And the groups that called the meeting were their respective scientific organizations, the national academies in this country. So there is an effort to do that. UNESCO has issued a statement discouraging, for now, doing embryo research with clinical applications. But it's just a statement. It doesn't have force. They want the international community to get together and work out these guidelines as well. But clear, not only international implications, there's going to be money in this research. It has economic side as well. And so the involvement internationally will not only talk about ethics and whether we shall go forward, it's also going to be influenced by changes that are happening in science everywhere in terms of additional considerations.
I'm Ruth Schwartz-Cowan from Irvington, New York. I have a specific question about the meeting in December and about the Chinese research that stimulated the meeting. If I understand it correctly, the research was done to deal with a very particular single-gene recessive disease.
Yes.
And I wonder, I know a lot about that disease, and it has a very large number of both national and international patient associations. Were the patient associations consulted?
Yes. The meeting was attended by bioethicists, mostly patient groups in December. And the national academies have created a committee to examine these issues. And the committee is taking testimony or listening to patient groups, individuals, and those who are concerned about these issues. I think all of those activities are terrific. My point is we need to go beyond that. And one of the reasons we need to go beyond that is that this is going to be fiercely contested work, and you want to have buy-in. The picture is not always good with respect to some groups and genetic advances. It's good with respect to others. African Americans, because this is one of the issues I care about, often gain as much as they lose by the time we finish talking about beneficial therapies that are going to come to our benefit, and we end up with additional stigma in other ways. That's what I would have liked to have talked about today. But I think I'm more voices, the better. And I think that people are interested in therapies. I don't know about enhancement, but clearly they are interested in therapies. And I'm not suggesting that the research not go forward. I'm suggesting that we are talking about the nature of human beings, as I said in the beginning, and we're all potentially affected.
Okay, I've told we're out of time. Thank you very much, Dr. Kang.